5-day meeting and then the travel, where it was going to be kind of crazy. It's a pretty long meeting, but they really wanted they made stuff. I haven't heard anything about it, and I have to deal with the times that we totally ridiculous. It's not like telecom, they just do it every day and make it fit. It's all day long. Ben is doing quite a bit. I got training. I don't know if so Ben has two topics, but he could send you a copy.
Thank you all for coming, especially our outside guests that are here today. We're welcoming you. And our speaker today is Professor Jeff Smith from Colorado State University, Pueblo campus. Jeff did his undergraduate work at the University of Minnesota in Duluth, and then took a PhD in biomedical sciences at the University of New Mexico School of Medicine in Albuquerque. He had postdoctoral training at Colorado State University in Fort Collins, University of Melbourne School of Medicine in Melbourne, and the University of British Columbia in zoology. So he is very broadly trained and has undertaken a fascinating new research program that is funded by the state of Colorado and a special set of 4 grants to the CSU Pueblo campus, and has agreed to come and give a talk about his work with cannabidiol, one of the more important of the cannabinoids, and I'm trying to encourage people to come in. Please grab a chair and something to eat. I will let him tell you more about the work that he has done, and welcome also his postdoc Amy, who's often the spark plug to the real worker bees in the room, are postdocs and graduate students, and guys like me. So, yeah, welcome.
Thank you. Thanks for the invite. It's a pleasure to be here. I'm going to give a talk about the research that we do in the area of neuroscience and CBD, and I'm going to show you today some of our brand new data. Characterize everything I'll show you today as preliminary. I made a transition beginning maybe 2 years ago when we were approached actually by a local businessman in Pueblo with the idea of doing cannabis-related research. And my original thought was, I don't know. I've got my own projects in monocarboxylic acid metabolism and learning and memory, and wasn't super excited about it until I started to look into the biomedical literature in the neurosciences about the biochemistry and the biology of cannabinoids and what they do in the area of learning and memory. and it became immediately apparent that it's a very important science, particularly the endocannabinoid signaling system. And so I was attracted to it from a pure aesthetic point of view, I think, as a neuroscientist, let alone the social need. People are using cannabinoids in a variety of ways, and that's expanding greatly in our country, and it's been going on for a long time in the rest of the world. We're kind of quite a throwback for not researching it more vigorously. And I think there is great need to really study it and understand what it does. So I'm going to tell you about the work that we've been doing, the directions that we're moving in. I invite you to please stop me and ask questions at any point in time. I've got way more material here than I'm going to actually talk about. I'm going to try and talk about something called synaptic plasticity, which is the neuroscientist's understanding of how we learn and remember. I'm going to talk about neural circuitry, which I know very little about how it works, but I'll talk about it in some respects. I'm going to flavor my talk with a bit of an insight into Alzheimer's disease, where I did a fair bit of research on the amyloid beta hypothesis and the etiology of Alzheimer's disease at the University of Melbourne in Melbourne, Australia, where I was a postdoctoral fellow. There's a couple other little things that I do want to make sure that I hit on. But look, if you have questions while I'm going, please stop and ask. So I'm from Colorado State University in Pueblo. Primarily, I do teaching duties. I've run in our College of Science and Math what's been one of the bigger labs. Our labs are very small by your standards, and our ability to engage our research is quite hampered by our high teaching duties. Nevertheless, we've been able to do things and publish, and proud of what we do. So I'm Jeff, and Amy, who accompanied me, is sort of the person that runs the lab. And Adrian Sculptorino just came and joined us about 6 months ago as the first postdoctoral researcher at CSU Pueblo. And I'm going to show a lot of work from Jose, Native American fellow, who turns out to be quite bright, talented in the lab, and has done just about all of the electrophysiology that I'll show you today. And he's going to do a lot more. And Zach, who's helped with the behavioral work that Amy's been doing, which I'll show you. So you're all aware of Pavlov's dog, probably the classic experiments that showed that animals and humans can make an association between one thing and another. So in the Pavlov experiment, it was an association between a sound and food. And the dog would show that he had learned that association by salivating even after training in the absence of food. So without the food stimulus, when the dog heard the tone or the bell, it would still salivate, indicating that it expected the food. It had learned that the sound of the bell predicted that there would be a food reward. And so I'm going to show you work that we've done with that paradigm in rodents today. The Pavlovian learning is associative, right? That the animal associates one cue with another. And it comes in two forms in the literature, and this is kind of new to me. I've only been studying this for about a year and a half. What's called the delay paradigm and what's called the trace paradigm for Pavlovian conditioning. The delay paradigm is where the vast majority of the literature is. And so people who have studied learning and memory using Pavlovian conditioning, and we're working in the area of fear conditioning. So rather than doing a positive reward where we give food, we give a negative reward, which is a shock. And it's a mild shock to the animal's foot, so it doesn't really harm the animal, but it does rapidly learn that a tone which we play predicts that it's going to get this shock. And it's powerful. Powerful to the extent that it's a sort of a primo method to understand learning and memory, because the changes that it affects in memory and the demonstration of learning is profound and powerful enough that you can even go in and look at the architecture of the brain after learning paradigms and see changes in the synaptic conductivity, the synaptic morphology. So it is bold enough that you can see it. This delay kind of learning is sort of reflexive. It is in the central nervous system. It takes place with brain regions, particularly a region or fear condition called the amygdala. But it's rather reflexive. It seems like it mainly, or maybe even in some people's opinion, only involves the amygdala. There's another flavor of this learning called trace learning, which is more cognitive. And in the trace learning paradigm, which is just subtly different from the delay paradigm, other brain areas besides the amygdala become involved, including the hippocampus and the prefrontal cortex. And we think, or I think, it's a better model of disorders of learning and memory, like Alzheimer's disease, post-traumatic stress disorders, dementias, anxiety disorders, and maybe even perceived pain, which are all disorders that are very cognitive in their nature. If you've ever known somebody who's suffered from PTSD and talked to them about it, and I've got a very good friend in town who's a combat veteran Marine that fought in Vietnam quite vigorously and suffers from this, and we talk about it. We're close friends. I had a friend whose mom died with Alzheimer's disease, worked on Alzheimer's disease a fair bit. These are cognitive disorders. They involve real cognitive kinds of components. So we think that the trace, because of its more cognitive flavor, is a better mode to study. Prefrontal cortex, hippocampus, and amygdala are the brain regions involved. We're focusing right now on the amygdala and the hippocampus. So you're all aware of the, or I assume people are aware of the endocannabinoid signaling system in the brain, which is when you consume THC, or in the endocannabinoid system, just when you do normal brain activity. Excitatory neurotransmission causes the production of endocannabinoids, anandamide and arachidonoglycerol. Molecular structures are shown here, which are released at the synapse where neurons communicate. And it basically signals to the neuron when it's active to slow down. So I'm talking mainly about glutamatergic, but also GABAergic inhibitory synapses. If that doesn't mean anything to you, just synapses are points where neurons communicate with one another. One neuron, the presynaptic neuron, releases neurotransmitter onto the postsynaptic neuron, which responds. And that's how your brain communicates from cell to cell, from point A to point B, to form circuits. So the endocannabinoid signaling system through the cannabinoid 1 receptor puts the brakes on neural activity. When neural communication becomes too hot, too fast, too much, this system kicks in and slows it down. And that's important because neural signaling can be very powerful, and it can be even damaging. So in diseases like stroke, for example, neural signaling starts to run away and actually exacerbates the sort of damage that is done after an ischemic event, stroke, for example. I'm not going to talk about any of that. We're talking about, in our work, cannabidiol. Well, I wanted to say one other thing. THC, though, is an activator of CB1, right? So that's how THC from the cannabis plant works. Cannabidiol is the other cannabinoid that has been studied the most from the cannabis plant. People say it's non-psychotropic. Doesn't cause psychotic sorts of, or a high, basically. It's purported to be non-toxic. People can tolerate, and animals can tolerate very high doses of it without having adverse effects. Antipsychotic, anti-anxiety, anti-pain, maybe useful for preventing substance abuse disorders. So people that are addicted to opioids might find some relief from their addiction from this compound. Cannabidiol, this non-psychotropic component of cannabis. Cannabidiol works in a fundamentally different way than does THC. It doesn't seem to have any particular affinity for the CB1 receptor that THC activates. So it works through fundamentally different ways in the nervous system, in the central nervous system. And I'll show you some of those ways as we go and what we're thinking about.
What's the difference between the red and the black?
The red is the stuff that I consider to be more related to learning and memory, whereas the black, the anti-seizure, is seizure Is epilepsy a disorder of learning and memory? I think fundamentally, I would think not, although it can affect your ability to remember and learn just through the brain damage that it causes. Neuroprotective could be very important. We were talking about concussion, I think, a little bit earlier, brain damage. But I don't think it's direct as an inflammatory or an anti-inflammatory. I could be wrong about that. Directly involved in learning and memory, because there are things that we consider to be inflammatory agents that are very important for neural signaling for learning and memory to take place. So, maybe I could have turned that red. Anti-tumor, secondary. So to me, red is things that have a distinct component that could be investigated as involving the mechanisms of learning and memory. So we do this fear conditioning paradigm where we put a mouse into a cage, and the cage has a microphone in it, or not a microphone, a speaker that can make a sound. And so we play a tone that the mouse can hear. And then we shock the mouse with a mild foot shock. It's in the order of milliamps, so millions of an amp. Millions, thousands of an amp. Microamps would be millions, so thousands of an amp. And we compare the tone. So on the top here is the tone, or the duration of the tone in time, with the shock on the bottom trace. So they get a very brief shock, maybe a second, or half a second even, very small. But they respond to it. They sense it. They squeak and they jump around the cage a little bit. And after, maybe 5, 6, 7 repeats of pairing the tone and the shock, they remember. And the way we know that they remember is they freeze when they hear the tone, even in the absence of the shock. So we have a video recording system on this fear conditioning apparatus that allows us to measure their movement very precisely and determine whether or not they're freezing or not. If they're freezing, then we say they've learned this association when they freeze in response to the tone. So this is what the delay paradigm looks like, where the tone, which we call the conditioned stimulus or the conditioning stimulus, co-terminates with the shock, which is an unconditioned stimulus. That's just jargon. You don't need to really evoke that to understand it. This creates this delay paradigm, which is reflexive and mainly only involves the amygdala. This part of the brain that's associated with strong emotion, fear. I think of it as sort of a supercharger for memory. If your memory has an emotional content to it, then the amygdala is turned on and it provides input to other circuits in the brain that provide for learning and memory. So if you think about your memories, things that have an emotional content are powerful memories. Your limbic system, which includes the hippocampus and the amygdala, receives a lot of odorant inputs from things that you smell, from your olfactory system. A lot of people report powerful memories associated with particular odors, for example. But when there's this emotional kind of a content, your limbic system is turned on, your amygdala is turned on, and makes your memories stronger. In the trace learning paradigm, we play the tone, and then there's a gap called the trace interval of about 20 seconds. And then we shock the animal. And for reasons that aren't fully understood, other brain regions become necessary for this trace memory, for this association to be formed when you put that trace interval between the tone and the shock. So it's more complicated. They have to do a little bit more thinking, and it requires other brain regions like the prefrontal cortex and the hippocampus, as well as the amygdala, to make this memory of the fear. So we think it's a better cognitive model. We've been doing this experiment plus or minus cannabidiol, which we give interperitoneally to the animals. And then we've been going in and looking electrophysiologically at what cannabidiol does to the brain activity at the electrical level. And the work I'll show you today is looking at populations of synapses and measuring the strength of the synaptic communication and other little temporal aspects of it that I'll talk about later. We can also go on and look at the morphology. And so we've been doing some of that to see how the actual structure of the synapses and the more cellular morphology, at a little bit grosser level of the individual cells, changes in response to learning and in response to the presence or absence of CBD. So the experiment looks like this. We do 3 protocols, a pairing protocol, an unpaired protocol, and a non-conditioning protocol. In the paired protocol, which is the big one, where we're looking to see the associative learning, we play a tone, shown in the green there, and then there's this little trace interval, which you can see right here. And that trace interval is 20 seconds long, and then we shock them for a second. And then there's an intertrial interval of, what is it, 2 minutes? 2-minute intertrial interval, and then we repeat this little pairing between the tone and the shock. And Amy's done this work, and she's been doing it 7 times. And then that's it, they're training. And we can measure their freezing during this training episode, and we call that learning. We think of it as learning. So did they learn? Did they freeze during the training episode? The next day, we can come in and see, did they remember by just playing the tone and seeing if they froze. In the unpaired protocol, we play them the same amount of tone of the same duration with the same amount of gap, the same amount of shock, but we don't pair it in any logical way. It's just randomized. And so maybe they're going to freeze because they don't like to be shocked. In fact, they do. They learn about all of these contextual components, the shape of the cage, the color, the texture, the odor. And yeah, mice do have sort of a primitive color vision. Somebody looked at that slide at one point and said, you know, mice don't see color, they only see black and white. That's not accurate. We do this in a cage that we call context A. So it's the cage like the picture that I showed you with the bars on the bottom that shock it and then the walls. And we also do just a non-conditioning, which is just pure context. We put them in the cage and we play the tone and measure their freezing in response to that. No shock. When we do the memory test, we do it in a different cage, context B. It's actually the same cage, but we change it. We change the way it smells, we change the way it feels, the way it looks, so that we're trying to isolate all of these contextual items away from just the relationship between tone and shock and measure just that particular learning. We end up measuring all these kinds of learning when we do it. So here's an example of a mouse, a white mouse, called a Swiss Webster mouse in a shock cage. And, oops, let's see. I'm trying to think of how to actually, with this. Okay, so there the mouse is moving around. And the software through the video camera records the amount of movement. This isn't in real time, but when we play it back, it really shows you the graph of how much the animal is moving. And so they will rapidly go out of this sort of exploratory mode when you start to shock them and play the tone, and they'll huddle, they'll just freeze. And that's a measure of learning. And so this is a memory test that we did 24 hours after a training session, a single training session where we just did what I showed you with the trace training for 7 trials, 7 shocks. All we do is we put them in context B, we play the tone, we measure the amount of freezing during the tone, and we can measure the amount of freezing in between tones. And what you can see with these white mice is that the animals that were conditioned do in fact freeze, and that's both if we measure it up here with the tone, during the tone, or down here if we measure it between tones. Same thing happens in both places where we measure the data. And we could see that the animals do in fact freeze when they hear the tone and when they're put into context B. And the unpaired animals that had the randomized tone shocking, they also freeze. And it's the difference between the unpaired control and the conditioned control that represents the amount of associative learning between the tone and the shock. So there's clear associative learning. We were warned by experts in the field not to try to do this with white mice. They said, you're not going to get a good response. And if you look, you know, their level of freezing is really low, like 20% of the time. That could create a pretty high signal to noise ratio, making it difficult to do meaningful statistics and such on this. They're behaviorally a different animal. They're pretty jumpy, crawly, climby, they'll bite you if you let them. So aggressive and kind of powerful for mice, mighty mice maybe. And look at the unconditioned control. They don't freeze at all. They just explore. They're just running around. You play the tone. They don't care. You put them in the cage. They don't care. They're like honey badger. Okay, so CBD actually when present during training, and this is an important thing to understand, is that we injected them interperitoneally with CBD at 30 milligrams per 10 milligrams per milliliter and per kilogram of mouse, right? So that's an intermediate level of CBD to put in an animal for these kinds of studies. And we put it into them when we were training them, when we were shocking them. Other people have put it into the animal just before the recall test, 24 hours later. And the paradigm there that people are thinking of is take somebody who has post-traumatic stress disorder and they've got this aberrant memory that is causing difficulties in their life. So if we give them CBD, will that reduce that memory? And so you can give the CBD before the memory recall test, where you just play the tone and see if they freeze 24 hours or 48 hours later. And measure the effect of CBD on recall. We're giving CBD during training. So we're asking the question, fundamentally, does CBD interfere with the memory acquisition process? Okay? So that's different. So non-statistically, there was a trend anyway towards a decrease in freezing. So it looked like perhaps they don't remember as well when CBD is on board when they are trying to remember something.
So may I ask a question? So the difference between the two panels seems minor to me when I look at it.
Yeah, I don't even have to show it.
Right.
I could have just showed you the tone.
Yeah,
What happened?
but then if I read your x-axis, it says % freezing during tone.
Yeah, either during the
I would
tone.
expect that to be the case when they have learned, and the lower says % freezing during intertone periods.
I don't know. We're looking at all epochs of both the training and the learning, and just looking to see what we see. And I think the people that are better versed in these sort of behavioral studies would say, well, no, this is really what you should look at to interpret it, particularly this way. And really my understanding of the methodology is not that refined yet, so I'm just showing both.
But what is your expectation? How do you interpret this?
I interpret
what's
it-
actually shown there? So-
When they go into the context and they hear the tone, they freeze. So we measure the amount of freezing during the tone or between the tone. Both times they freeze. Maybe we could have just combined it and said when they're in the cage and we're playing the tone, or we could just say during the tone, or it doesn't seem to matter. They freeze. So
they freeze when they hear the tone, and they freeze when they don't hear the tone.
they're in this context B, remember, right? So when this is done, they're in the original cage, but it's modified in a way so it smells different. The floor feels different. It's a smooth floor instead of grids. And for these studies, we put a piece of cardboard around the sides, colored cardboard, that was actually a little bit problematic because they would jump over it and try and get behind it and climb around on it and stuff. But we made efforts to change the contextual environment so that all that really the animal should experience is the same tone. So when we start playing the tone and we put them in the cage, they freeze 24 hours later. And they freeze more than if they were only ever in the cage in the first place and heard the tone. And they freeze more than if they were in the cage and heard the tone and received the shock, but in no particular order. Right? So that's typically interpreted as a measure of Pavlovian fear learning. So they learned the association between the conditioned and the unconditioned stimulus.
But the freezing happens in a somewhat random way, is what I take away from this, that it is not correlated in time to the tone.
Depends on the animal a little bit when you look at that data and present it that way, and some people do present it that way. You do see some animals, particularly the black mice, I'll show you in a little bit, where during the tone they'll freeze more, and then in the intertone interval they'll freeze less and then more. But they're animals, they don't behave perfectly, and our N's are small. The N's in these studies is 6 in each group. So that was a total of 36 animals to do that. And we began to repeat it before we were going to attempt to do any statistics on it. And on the controls that had no CBD, so the blue data, we got a different relationship between the unpaired and the conditioned control. And we started to think, gee, Steve Reich was right. we shouldn't have been messing with these white mice. They're unpredictable, and they don't freeze much, and they don't do the same thing. The reason I chose white mice is the Nobel laureate in neuroscience Eric Kandel has published that the black mice, which most people use, are not an optimal animal to study long-term potentiation in brain slices with. And the white mice are much better. And so we had downstream applications planned that if we could get these white mice to freeze, it would have been ideal. We abandoned them when we moved to the black mice. I'll show you that in a minute.
So I have a question. So you have a total of 36 animals.
Yeah. 6 in each bar.
And then do you have a significant difference between animals?
I think this would be, I didn't, doing statistics on ends of 6 is a little
really
bit,
hard.
yeah, it's a little bit unbelievable. So the plan was to repeat this, and we would have 12 in each group, which would be pretty good for these kinds of studies. Better than most people do. Most people can at 8 or 10. But I'm going to say these are trends. Clearly, we could get some associative learning at least sometimes.
And then between those two blue bars, is there, between those two, is there a significant difference?
There would be.
There is.
But when we repeated it, it was dubious. It looked like it would be hard to get a small effect, and so we abandoned the white mice. I'm still showing you some white mouse data. It's interesting, some of the geneticists might wonder, I wonder what the genetic difference would be that would account for this. Or behaviorally, you could analyze it, and you could say, well, there are these clear behavioral differences between these really aggressive white mice and more docile, kind of passive gray mice, or black mice. And so you could at least say, well, these effects, this learning and the effect of cannabidiol, as you'll see, depends on the sort of behavioral background of the animal. And we thought we even noticed among the white mice that were more docile just by their personal nature seemed to respond differently than the more aggressive white mice. So doing these kinds of studies with behavior, you've got a whole system intact in a living animal. It's really complex. It's pretty unpredictable. So to me, it's remarkable you can get anything that makes sense. This was during learning, so we could measure their freezing during learning. And what we saw, which was interesting, was that in the presence of cannabidiol, they froze more during the training. So during training, they seemed more responsive. So I attended a meeting where I met a number of people who use cannabidiol for various maladies and or whatever purpose. And I queried them about it. And one person said to me, it's like taking a television set, and if you had a button to go from low def to high def, it turns on sort of this heightened sort of sensory perception. And it's consistent with what we saw. They seemed like they were much more tuned in to what was going on during the training. Anyway, we gave it up. We went to these black mice. These are still just ends of 6, although as we speak, Zach Montoya should be beginning the fear conditioning experiments for the second set of 6 for the non-cannabidiol. Next week we'll have finished the other 6 for the cannabidiol animals. We'll have ends of 12. And it's more robust. Right away you can see more freezing. This is during learning. This is during the training intervals. When we play the tone, there's a trace interval, shock, intertrial interval, repeat 7 times. When we measured their freezing during the tone, during the trace interval, or during the intertrial interval, what you can quite clearly see is the same thing in the black mice. In the presence of cannabidiol, they freeze a lot more. So I wouldn't say that means they're learning better, but it seems like they're freezing more. So it seems like they're more tuned into the sensations that they're receiving, and cannabidiol is clearly enhancing that. We're also getting actually pretty comparable levels of freezing in the black mice that are the control animals. A little bit better though, a little bit better, but a lot better in the presence of CBD. The memory test is the one that is really interesting. When they've got cannabidiol on board and they learn, do they remember? And 24 hours later with these mice, we saw a dramatic increase in freezing in the presence of cannabidiol during the learning trials. So that really surprised me. That led me to think, well, maybe it's a memory-enhancing agent. We actually had hypothesized that because some of the previous data published in the literature showed that doing extinction training, which is kind of what we embark on during the learning, where we play the tone and measure in the absence of shock the amount of freezing. If you keep doing that, it's called extinction training. It's akin to exposure therapy for people with phobias and such, PTSD, where you expose them to the stimulus that they had associated with the pain of the shock, and you never give them the shock, and pretty soon they relearn. They learn something new. So it's not forgetting, it's actually learning something new. And when you do this and give them cannabidiol, the mice cannabidiol, during the extinction training, they actually make this new memory that the tone no longer predicts shock more robustly. So to me, as a guy that thinks about learning and memory, it makes me think that it enhanced their ability to learn and remember. So we had actually had my undergraduate student hypothesize that it would enhance learning. Now, a lot of other literature done with the delay paradigm and done when they bring the CBD on board during or just before the memory recall test suggests that it would be inhibitory to learning and memory. So I actually kind of quietly thought this is going to inhibit learning and memory, and when the white mouse trend showed up, I said, aha, I knew it. It's bad for learning and memory. I'm still kind of processing this. This is brand new. But take a combat veteran like my friend who was a Marine in Vietnam, and say he's doing exposure therapy to try and work through his post-traumatic stress disorder. Cannabidiol, some of the other literature, I didn't show you any of this today, but things that are published would suggest that that exposure therapy, that therapy would go better with CBD on board. However, that soldier might, if still active, take that and extrapolate it to the CBD helped me with my therapy, so I'm going to take it out on the field. This data would suggest that out on the field it could actually enhance the acquisition of the fear memories. Right? So to me it underscores, it's a clear example of why we need to do this sort of research, so that we can get the context straight, so that we know what is an efficacious, useful, helpful application, what could potentially be harmful and should be avoided. That's probably our most exciting data. And I spent a lot of time on it, a lot more than I intended to. I've got other electrophysiological data that I'll show you. I'll probably have to, what time am I done? I got about 20 minutes probably at most.
Right.
So I could talk a lot, and I've got enough data here, slides, to show you, we could talk for 3 more hours. I could stand here and go through this. I'll try and just make some main points. Stop me if I'm not making sense. This is a picture of a brain slice from our lab. It was Golgi stained. It shows some of the neural architecture of individual brain cells. You zoom in on it, you can actually see their dendritic spines, which are the postsynaptic processes that form the postsynaptic part of the synapse and neural connections. This is some electron microscopy that has been published through computer-generated serial reconstructions. It's published in the literature, and people use this technique now, showing some of the ultra-structured molecular distribution of postsynaptic proteins and such at these dendritic spines. The spines exhibit something called plasticity. In fact, the entire synapse exhibits something called plasticity, which means that it can become much bigger and stronger in its ability to communicate between the two neurons, and it can also become much weaker. So that's called synaptic plasticity. So when you learn and remember, if you think of your brain as having neurons that are interconnected at all of these thousands of synaptic connections, both pre and post-synaptic, the wiring diagram, this is a picture from Ramon K. Hall back at the early part of the 20th century. He drew this from his early microscopy, famous work, showing some of the neural circuitry in the rodent hippocampus, an area that's well known to be important for the acquisition and storage of memories, particularly spatial memories, but also emotional memories. You can think of this circuitry as being like the circuitry in a computer board, right in a circuit board in your computer. Your circuit boards in your computers and your phones, they take an input, they process it, and they give an output. Stereotypical. Same thing happens every time. This kind of data goes in, processes it by virtue of the interconnections of the circuit elements in the circuit board, and then it generates an output. Well, your brain circuitry does the exact same thing. The neurons are connected to each other in a precise way. But plasticity causes that circuit to be able to change in real time in your brain, which that doesn't happen in your computer circuits, in your phones and computers. So that gives our brain considerably more power as a learning and memory tool. Here's a synapse. Yeah.
What's the scope of this plasticity? Like, is there an absolute cap on some kind?
I've seen in even in vivo studies, sometimes the strength of the synapses appears to be maybe twice as strong and can be diminished entirely. So you can have circuit elements becoming much stronger or going away entirely. And it all happens in response to the pattern of communication. So a typical neuron, I tell my students in my anatomy class, it's like a voting machine. A typical neuron connects to in the hippocampus, an excitatory pyramidal neuron will connect to 1,000 other neurons. So it's like if I was a neuron, I have an output to maybe 2 or 3 other neurons, maybe you and you and you. Okay? And I have inputs from everybody else, and each one of you would represent something like 100 different synapses, right, or 50. And so if you release neurotransmitter onto my dendritic tree, that might not be enough to get me to do anything. I won't communicate. But if you and you do at the same time, particularly, that might cause me to reach a threshold to do my job in communicating with these other neurons. So if you do this very rapidly, and it's frequency dependent, you have a higher ability to cause me to our connection to become stronger. Whereas if you're communicating with me really slow, like 1 hertz, then a pyramidal neuron in the hippocampus, that particular synapse will get smaller and smaller and smaller until it goes away. But if you're communicating with me at 100 hertz, that's a pretty good frequency to make our connection stronger, stronger, stronger. So in the grand scheme of the wiring diagram of these different brain regions, plasticity plays an infinitely complex role, difficult to decipher, in modifying the circuit diagram of these brain regions that process inputs and generate outputs, sensory inputs and motor outputs or other outputs to other brain areas that are cognitive in their function. All these different proteins in the dendritic spine, a really remarkable structure. Eustace, his book, he describes it as a molecular crystal, just almost void of water. It's so full of proteins, actin and signaling proteins, and receptors, trafficking vesicles to modify the structure of these spines. You can actually see them in vivo. Studies have been done where you can induce potentiation of the synapse and look at a single synapse with 2-photon microscopy. In about 4 minutes, if you give it the right high-frequency stimulus, it will poof, get much bigger, get stronger. So the neuronal firing patterns, the pattern by which the presynaptic neuron releases neurotransmitter on the postsynaptic neuron, which responds, that frequency of communication between those neurons determines how that particular circuit element changes. The rewiring is required for memory acquisition. Plasticity is important. It's necessary for memory acquisition. If you don't get strengthening of certain circuit elements during a memory acquisition, you don't get acquisition. So if you block synaptic plasticity, you block learning. You also block the persistence of memory if you block synaptic plasticity. Did I make that clear? So this rewiring, this restructuring of individual synapses is required for learning and for memory. And it involves signaling among all of these proteins that are present in the synaptic apparatus. particularly important protein is the NMDA receptor. I'm not going to go into it because that would take the rest of the time. But we can understand through the mechanism by which the NMDA type of a glutamate receptor functions, we can understand why it seems like the trace memory would require something called endogenous persistent neuronal firing to bridge this trace interval. I can't explain that to you in 10 more minutes and give you a coherent rest of the story. But if you take it at face value that because of the way the NMDA receptor works, I think that the way that the trace interval brings on these other brain regions and functions for trace memory acquisition potentially involves this thing called endogenous persistent firing. And I think cannabidiol can modify that. Really want to talk about this, but I spent too much time. Here's a pyramidal neuron, and it shows all of those dots represent synaptic connections on the dendritic arbor of that particular cell. Just as a picture of, a real picture, it's immunofluorescence of a real cell, showing what I was just describing about the thousands of connections to one neuron. And then it's, one of these is its axon that connects to just a few other cells. There's a difference if you think about it in computers. I've understood this by thinking about computers. A computer circuit board, like I've been talking about, processes information by virtue of its pattern of interconnectivity of the wires in the circuit board, the different circuit elements in the circuit board. But memory is fundamentally stored in a different way. It's stored on a hard drive as magnetic charge distributed at particular loci, plus or minus here, plus or minus here, plus or minus. It's fundamentally different. We don't fully understand how memory is stored. We don't fully understand how memory is acquired. But it makes sense that the acquisition and storage involve some differences in the process, at least by analogy to computers. Eric Kandel, working with snails, has advocated the idea, though, that no, it's the rewiring. There are enough synapses in your brains, more than there are stars in each of your brains, to account for storage of memory by just rewiring particular circuits. Okay, I think it's probably a mix between the two things I'm thinking, but that's just my bias or my dumb opinion, really. Kandel would suggest that what happens is that the rewiring becomes persistent and it stays, and that's what holds your memory. Okay, a lot of the fear work, where we've gone in and actually looked at, sort of statistically, at the changes that happen in synaptic strength during fear learning, has strikingly demonstrated that the synaptic plastic effects, the plasticity effects, only last for about 10 days, and then it kind of goes back. So to me that makes me think, well, it can't just be persistence in the rewiring that stores the memory. Somewhere else the memories are distributed in some other way. So regardless, there are these proteins called prion proteins, or prion-like proteins. Amyloid beta is a prion-like protein. The prion protein is the kind of canonical prion protein. My mentor at the University of Melbourne, Colin Masters, was a leading expert in prion protein theory. I had the privilege of working with him. So Kandel thinks, first of all, I want to tell you that when memory is stored, it requires protein synthesis. Proteins have to be synthesized, and synaptic proteins are known to be increased in their synthesis. So the level of these synaptic proteins goes up during the storage of a memory. This includes things like actin, which is a protein that forms the skeletal structure, sort of the scaffolding in the spine. Receptors for neurotransmitters, signaling proteins. All of these things are upregulated during learning, right, as you get synaptic enhancement. Storage, which is regulated by modulatory neurotransmitters like serotonin and acetylcholine, which I hope to be able to tell you just a little bit more about what I'm thinking about that as it relates to this endogenous persistent firing. What we think, though, besides this modulation by these other neurotransmitter systems, is that activity and the neuromodulation tags an active synapse by causing them to increase the expression of prion proteins. So a synapse that undergoes synaptic plasticity increases its level of a prion protein. Prion proteins were sort of an enigma for a long time because it was discovered that they could catalyze their own formation as a protein. It goes against what's called the central dogma of molecular biology, that DNA makes an RNA and RNA makes a protein. That's the way. With prion proteins, it looked like no protein makes protein. Right? What's called the central dogma of molecular biology. Molecular biology doesn't really have a place for that. So it was sort of enigmatic. But when you have a prion protein, it can exist in two forms, a normal, healthy form and a scrapie-like form. This protein can cause things like mad cow disease or Jacobs-Crutchfeld disease. I'm not sure I'm saying that right, but when the prion protein undergoes a conformational transformation, it can then catalyze that conformational transformation among other normal prion proteins. So what Candel has some evidence for in aplasia is that these prion proteins aggregate and trigger the transformation of other prion proteins into this scrapie-like form in these localities of these individual synapses, and stimulates ongoing protein synthesis that keeps these particular synapses strong to keep that rewired part of the brain rewired. I think it's very interesting in the context of Alzheimer's disease, where for unknown reasons, the amyloid beta prion protein, which is a ubiquitous protein throughout your body and also synaptic, suddenly becomes expressed and starts to aggregate in much greater ways than it normally does. And it's absolutely associated with the etiology of Alzheimer's disease. So, exactly the role these prion-like proteins are playing in the brain at the biochemical level we're still studying that a lot in the neuroscience world. Clearly, the prion proteins are important for memory storage. THC has been shown to reduce the expression of amyloid beta from neurons in culture. That was just shown, wasn't it the Salk Institute that showed that? Right. I'm going to show you data here really briefly that cannabidiol can clearly modulate neuronal excitability, which is involved in the production of these prion proteins and memory storage. These are LTP studies, long-term potentiation. That's a mouse hippocampal brain slice that can record a waveform that can measure increases in synaptic strength that persist for over an hour, called a long-term potentiation assay. So that's a picture of my lab in Pueblo. We make these measurements of synaptic plasticity in living brain tissue from mice. So here's some studies that we just did in white mice, and we're stimulating and recording in various regions of the mouse hippocampal brain slice. And we see a baseline response, and then in the presence of CBD, you can tell that response was diminished. And that response basically measures the strength of synaptic communication. And when we take CBD off the slice, just wash it out, it returns right back to baseline. So it's fully reversible in the hippocampus. We also look at something called the paired pulse ratio, where we do two of these measurements in rapid succession, about 50 milliseconds apart. And because of calcium dynamics that control neurotransmitter release in the presynaptic neuron, we're actually studying something about presynaptic calcium function and how that relates to neurotransmission. The paired pulse ratio is a measure of this. And we saw either no effect or maybe a small inhibitory effect with the white mice, enough to just about give it up. But with the black mice, the paired pulse ratio showed a dose effect, micromolar, 5 micromolar, 10 micromolar, and 20 micromolar CBD. And the paired pulse ratio is decreased by about 10%. So presynaptic function, as others have shown, is clearly affected by cannabidiol in this system. And these are reasonable doses. 10 micromolar is probably achievable in a living organism like our mice that have received the group paratin. even more interesting to me, or also very interesting, if you look at just that little part that I circled, it's called the fiber volley. It's a measure of the action potentials in the neurons that we stimulate in this experiment. And you can see clearly that in the presence of CBD, it takes longer to get from point A to point B, and then it's reversible. If you think about the wiring diagram of the hippocampus, small changes like a half of a millisecond could add up, and with all of the different circuit elements interacting, could, I think, at least cause some sort of a distortion of normal hippocampal processing. In the black mice, we see this decrease in the action potential as time course with a dose effect. I express this in milliseconds per millimeter, so about a half a millisecond at most per millimeter of axonal path length. So for the neural signal to get from point A to point B, it's getting there slower in the hippocampus of these mice. We think it might have something to do with sodium channels. Looks sort of like an anesthetic effect. This is amygdala. Just started doing this last week, so these are really low ends of 3. Here's the hippocampus in a brain slice. Here's the amygdala. So this is where I stimulate and record if that means anything, but it's the amygdala. And people make this measurement, and you get this waveform. And you can see that CBD really reduces the intensity of the response, so the neurotransmission is not as strong, and it also delays it a little bit. That's just a sort of a dose response for the strength of the stimulus. It's not reversible. It's just ends of 3. You've got a lot more work to do here, but it's very interesting. I told you it was all sort of preliminary data. And this is the lag, the delay in the amygdala, and that's also not reversible. So is this an artifact of my experimentation? For some reason it doesn't wash out in the amygdala and it does in the hippocampus. I'm still looking. Lots to be done to make this data, all of this data, of high quality. I'll just finish in about 2 minutes. Acetylcholine receptors and TRP channels have an interrelationship. Your forebrain projects neurons that release acetylcholine into areas like your hippocampus and your amygdala, and they modulate, they're neuromodulatory. So they change the dynamics, the strength, the physiology of neurotransmission in these brain regions when you function. Acetylcholine neurons degenerate in Alzheimer's disease. And in fact, I'll just skip to my last slide. Cholinesterase inhibitors like donzapil are compounds that have been used with modest efficacy in Alzheimer's patients by increasing the hormone of acetylcholine neurotransmission. The way I think that works, what we're kind of chasing as our hypothesis, is based on this idea that, first of all, I see that didn't look like it came out too good, but cholinergic signaling is really important for normal learning and memory to take place. If you look at it in brain slices at the synaptic plasticity level, or if you look at it during learning in whole animal systems, cholinergic system is important for learning. CBD has a powerful inhibitory effect on cholinergic activity, at least in some very simple experiments. Cholinergic signaling through a fundamentally different kind of acetylcholine receptor can activate this thing called endogenous persistent firing. And this endogenous persistent firing of a neuron that just once it's turned on by cholinergic signaling keeps firing action potentials. That tonic, continual, persistent firing of action potentials, I believe, could be, and I'm not the only one, necessary for bridging the trace interval in the trace learning paradigm that I described. So if CBD can modify cholinergic signaling to change endogenous persistent firing of neurons, that could explain its effect on trace learning in our mice. And so that's kind of a little bit further down the field. Hopefully by the end of the summer we'll have emerged with some data. We're working with things like transgenes that we're just expressing locally in individual cells to see if we can find an effect on these channels with CBD. seems like, as always, I wanted to tell you a lot more than I really had time to tell you, but I hope at least I gave you somewhat of a picture of where we're going, what we're thinking, and what our future holds. We will be having a first annual conference of the Institute of Cannabis Research at CSU Pueblo in the end of April. You can look that up. Just do a search for Institute of Cannabis Research or just cannabis on the CSU Pueblo website. You can find it. I invite you to come and participate as in whatever capacity if you are interested. You can learn more about that then. Okay, thanks.
Time for a couple of questions. Yes.
What was your delivery system?
Interperitoneally in ethanol and tweeting. It's a pretty standard way that people inject this. We're not doing anything like interventricular or localized injection. It's just interperitoneal. Yeah. But what's unique about our work is that we're giving it just prior to the acquisition rather than during the recall for testing.
So the amount, I just want to know how realistic that is. I missed the last part of the talk, so I don't know if you talked about it. But so you injected. Mm-hmm. The amount that you inject, is that kind of comparable to what someone would smoke? First, and second, could your results be extrapolated to another way of acquiring CBD, like smoking it or vaping it or eating it?
My somewhat rough understanding of the literature, and Amy will correct me if I'm wrong, is 30 milligrams per mil per kilogram is probably on the high end, and we used 10. And I think people see effects down around maybe 1. And then effects aren't as notable after that. My dose response curves there on the brain slice stuff at the micromolar level, 20 micromolar is probably the upper limit that can be achieved in vivo by whatever delivery mechanism. So where I'm still seeing kind of the high end of the dose response at a micromolar, to me is really encouraging. To me that's a lead. And it's congruent since it has this kind of anesthetic effect. Very congruent with the data out there that supports the idea of CBD being very important as being a helper for people with seizures. Right? If it slows down neural activity, that could be good in a seizure paradigm.
Has there ever been any studies or any hope for between connecting the severity and frequency of the PTSD and like Vietnam vets? Or those who are smoking pot regularly, those who won't, or is it just too many variables?
I don't know. That would be a really interesting study. I know my friend uses it, and I think my sense is he uses it to his detriment. I would say he doesn't use it; he abuses it. But
and I actually was wondering about the learning phase, like during Vietnam.
So, I didn't go to Vietnam, but I watched movies and I talked to him, and it sounds like people were smoking it over there during combat.
I've seen those movies too.
So, but, to me, that's maybe the best thing I had to say today was that my data seems to support the idea that having cannabinoids on board during some contexts could be harmful, and in other contexts could be helpful. And so I think understanding it by doing these sorts of studies helps us to know when and how to use it and when not to use it. And that's an important part of any medicine. I mean, I always tell my students, and you'll get it too, is when you hear the advertisements from Big Pharma for different drugs, what is the ad about? It's about all of the off-target harmful side effects. And it takes R&D to understand that. R&D that I don't think is done for the vast majority of drugs. So.
How would you compare the likelihood of success of actual neuroregeneration damage as opposed to neuroprotection?
So, free radical types of things like superoxide, nitric oxide, which works in a different way, but superoxide is pretty interesting because superoxide production is David Sweat showed some of this work in his LTP studies, that superoxide is actually a free radical, highly oxidative, potentially very damaging species that is important as a signaling molecule for synaptic strengthening that normally should happen during learning, or at least that's sort of the theory. So, when we think about take an antioxidant, that'll solve your problems because oxidative species are absolutely damaging and harmful. If that's your viewpoint, it's probably not accurate. So cannabidiol has antioxidant properties, which could be good when oxidative damage gets out of hand. And so the endocannabinoid system, remember, puts the brakes on neural signaling when neural signaling gets too high. That could potentially prevent the generation of oxidative species that go overboard and signal cascades that lead to cell damage or death. Whereas if you have in an anti-oxidative or anti-inflammatory kind of capacity, that can be helpful in some situations. But for normal learning and memory, could it be that I would find that memory inhibiting functions of some compound are mediated by reducing oxidative species? That's entirely possible. I kind of thought there for a time when I saw that the cannabidiol in the white mice was enhancing their freezing response during acquisition and somewhat inhibiting it during the recall test. I thought about pro football players or boxers. I saw a really good boxing match with my son on TV on Sunday or Saturday night. And, looking at these guys take this abuse, if that's the way it works, like in my white mice, then you might say, well, cannabidiol might be a pretty good performance-enhancing drug for a football player or a boxer to take during the performance. Now, of course, that comes with all kinds of ethical questions, but if it then has anti-inflammatory, anti-damaging effects later, it could also prevent some of that downstream damage that happens after. So, maybe that's a useful context for a drug like cannabidiol, but now I see with these black mice something entirely different happening, something that I'd originally predicted. So it's very complicated. It's hard to... And, people that you talk to, I talked to an older lady who said, I used cannabidiol for, I can't remember what she said she used it for, but she said, I quit. And she said, I don't know why they say it's antipsychotic, or why it doesn't have psychotropic effects. She said, for me, I get very tired. I couldn't drive. She said, plus I fell asleep when I would use it. Whereas this other guy said, no, it's just like turning on the high-definition television set. All of a sudden I see the world crisper and sharper. So these two people standing side by side are reporting, disparate sorts of experiences.
All right, last question, man.
So you're using a synthetically derived CBD, is that correct?
We're using cannabidiol. We purchased it from Sigma as an analytical standard in methanol, and then resuspended it in ethanol after evaporating it under liquid nitrogen at 70 degrees. We also have some crystalline CBD that we're using in ethanol, 100% ethanol. So it's actually from him.
Okay, let's thank our speaker again. The meeting in Pueblo should be fascinating. Yes. And so go to it. It's going to be worth it. We're good. What I'd like to do is kind of see if
Yeah.
Yeah, I'll do that. Does that answer? I mean, that answers.
Ideally, our students would say, oh gosh.
Hey, buddy, how are you?
Regardless of whether I see it on an exam, it's always what we do.
Whatever. We can do.
Yeah, but I met with some
We're definitely into it.
So I was just trying to dissolve in my mind. Third literature studies are response from the CBD.
So our farm is Durango.
And so they've also done the learning using not trace. It was not a trace.
Yes.
We're looking at other cannabinoids as
So just
well.
trying to kind of resolve that all with the last results you showed. You had an opportunity to explain today.
I got a story of that.
You have to trace
So I would plan to put all
all
this
the
from
inhibit.
They create these stuff.
For things that are immediately the same, right? So with the
chemistry in the past.
Yeah, cool.
The other thing we figured out was, we put it in
that happened.
Okay, so I kind of take, my take on this, it has the potential, but I don't know, to modify the timing of that surgery so that it does.
Yes, that was reach out and hang out with the scope guy, send you some info. It's good to see you. Yeah, thanks for staying in touch.
Hey, have you met Zeno before? I
Yeah.
don't know if you guys have had a chance to meet. Zeno works with us, he's our engineer.
Oh, cool.
And so I bet he'd love to see your trailer sometime.
Yeah.
Oh, you
We're getting out the trailer.
Yeah, I've seen it.
Yeah, it's really impressive.
And so his plasticity is being
Well, so yeah, for glasses is but I had my choice to do it all over again. However, though, when you're doing sort of research, it's really nice to see everything. Yeah, you want to see it all. And so I just run it. It's fine. But in order to get throughput, I'd much rather have a larger stainless operation. And I've been researching that, but they get really pricey. In fact, all this equipment is incredible. As you guys probably know. And you can blow a budget like one item.
We were just thinking about that this morning. Yeah. You could close the university in 5 minutes.
I know, exactly. So yeah, nice to meet you. Take care. Yeah, maybe sometime I'll either bring it over or you can come on out.
All right, so the hazardous waste disposal meeting is tomorrow.
Bob, this is my wife's group.
Could you do me a favor and just do the inventory sheet on it for me? And
So they're gonna
I'll
come
pick it up tomorrow. No, I'm gonna take it over there.
And then,
So you check out what time it is. Yeah. So just
Yeah,
do
yeah, no, we love the work you're doing.
the lab tomorrow, or you?
Yeah, I mean, really, it's
I just
No, it's
started
just,
on
I just wanted to
that.
get the three of us together, just to, I wanted to brainstorm about, a little bit more about, well, I don't have the answer to the Cameron yet, so I was just looking to have it. Start making sure I haven't heard from him yet.
All right, nice to see you. See you.
yeah.
but
We do.
we
We do.
no, I got the same right now.
Yeah.
Realistically, this company wants to certain, you know, I have.
Hi, my name is Rich.
I'm gonna drive
Hi,
separate.
They
nice to
kind
meet
of
you.
want
That's
to
fine.
know that I'm gonna spend
Oh, you are? Right.
Yeah,
like,
with no
Right.
like, and other times it's like
Yeah, so I met Bob.
I didn't know
I got
exactly, I
a
know what I
position
have
developed
to do,
farm.
but exactly how I have to shape it or do it.
Yeah, well, all kinds- by the way,
That's
you
what I
have just-
need, like, so it's
Oh,
like
boy.
trying
You
to
do what you
Doesn't
need to
strain
do.
for
I just was
another
responding
strain.
to that comment you had made that you
Yeah.
felt like he had overcharged.
And
But I was
he
gonna
got
say.
really good connector.
No, that's all.
Same way as you do.
That was last month.
So
we got
we're
announced
doing a
that
lot
at
of,
another guy's office, and he had
you know, just
where's
breeding
money
projects,
from? Where's the
trying
charge?
to breed
This guy
for
charged
specific
4 hours,
cannabinoids.
No.
Males
I-
and females, but
I
I
try
mean,
not
what
to
we
be,
do
because I know
is
it's like,
we obviously breed
know
for seed,
that
and
there are
then I
things, you know,
do
Right, you have to pay
we're,
attention, right.
propagate early seeds and then do analysis on those. On each one.
cannabinoids. So what, so how
Right,
do you work with cannabinoids?
Yeah, no, I.
So the males, usually we start them someplace else, and then we're going to feed them with our greenhouse, and we basically we process everything in the greenhouse.
The outside door would be great.
And we'll do a run that way. So,
Left or right? Oh, it's a ways away.
Yeah, they're gonna take this to.
No, I just slammed one.
I got my children place.
So I
No, I totally get it. No, you do what you need to do with that. I just don't want you to be like.
work.
No, but the other side is I just want to make sure
Yeah.
that what they're
And
used
we
to.
have these
Yeah.
They're used to
interesting
just
seeds
catching up to accounts.
that whose genealogy we know, so we know who our grandparents are, we have secrets from the grandparents.
Email me and I'll give you all
And
the
the thing is that,
information.
Sounds good. I will
Thank you so much for your talk. That was
and we
very
have not been
helpful.
able to find a place where we can have males, where we can
All right, right.
distribute. And I'm
So,
sure-
are you in federal?
That was
Yeah, I'll see
fascinating.
you in-
Well, go ahead.
I see you. I am here.
Then I'm
Oh, you're
like,
both.
oh,
Yeah,
Okay.
you are there.
exactly, exactly.
So we are, you know, one of the reasons why
So-
having males is
But yeah, all I
not
was
such a
saying
big
is
deal for us is
I
because
have to do a little
there
bit
are
of
no
that,
farms near
and
us
I just wanna
for
make sure that
50 some miles away.
Yeah. And
So
I think
we're
you've
like
done that.
right next
I
to
think
the
you've done
wilderness
that.
area.
So here's the, all I was.
Which is
gonna
good
do
and
about
bad. I
that
mean,
was-
the good part of
was
it
I
is
was
we don't get
hoping
the kind of
we were gonna
pests,
have a check
we
again,
don't
and I
get
think it's
security
coming
problems
this
with
week.
people running through
And
there
then
all
the
the time.
second thing is we've
So
got that
that's
call
why
on
we wanted
Friday
to turn
with
it
that
into sort of
inhaler company. And so
research on
this guy
breeding and
is kind of
cultivars.
bugging me because he's like,
So-
send me things. I'm like, "Oh,
Hi, yeah.
you know, so
Yeah.
I just wanted
We met
us
before.
to brainstorm a
Yeah,
little
okay.
bit, just
You
the
came out
three
to
of
my
us
place.
about like, okay,
yes.
so
Right,
we
okay.
want to,
So, are you not,
you know,
so
I'm
will
looking
your
at
farm
the cost
still
of house
in
In Durango,
the
yeah.
cost
yeah.
of like all the
Yeah.
different options of what we have and what's
Oh,
realistic.
that's 6 hours
And how
away.
you scale
Yeah.
something like this up.
Yeah, what we do is
Their,
we bring out samples and then we do the testing
equipment
here and
to
then we
use
do
their
extraction
inhaler if
too.
we even could use
But
it without
we don't do it on big
like
scale. We're
huge
small,
licensing fee.
sort of
And
R&D
then we
scale.
need some specialized
Yeah.
like production
So,
line in order to fill
I've
it
been
because it's
talking
in
to
this
other
tape.
people,
But if they
I
offer
need
all
this
those,
is a-
I don't
if
need
they
to
offer
grow
all
many
that,
nails, I
you
need
know,
to grow
the filling and all that, and
Yeah.
cost comes up.
because
My question
That's the way to go.
Right,
I
my
can
question is
explain
like, we really,
later
like,
how
if it's
all
more
the
than.
process was,
we
but
can
we
discuss
had
whatever we make.
these
Right.
Then it
males,
comes down to
females that
the
we
design
crossed,
of that
and
stuff
they
is meant for
produced
people to
a,
go with the
you know, like
compromise.
offspring,
Yeah.
and from
Yeah.
one
And it's
of the
great,
female
but
Yeah.
offspring, we crossed back
Yeah.
to
We
the
don't
dad,
say
and
back
so
cross.
on what we
Yeah.
want to do,
Exactly.
so we
But if we
have
want to have
actually,
a
yeah, we did a back cross,
Right.
and from
then
a
we
back
do
cross
want that.
population,
Those are
Right, right.
Or
we have
multi-dose.
13
Multi-dose
females
is the
Right.
big
that
thing,
we
I
crossed
think,
from
in
high
this, and the
school.
But I think there's cheaper
convenience.
So
Right.
we have, so these individuals who have the
Yeah.
seeds,
We can use,
and
yeah.
these seeds,
Yeah.
Just need to get one.
they are more
Exactly.
Yeah, exactly.
I'm China, and I
than
really
brothers and
consider
sisters
all those
because of
companies
the
on
increase
Alibaba
that we
say,
have. But we
do
want
it.
to keep on integrating them.
So that being
because
said,
we want to make,
right now,
so we're trying to make a genetic map.
to do
A genetic map is a
200
tool that
pounds
tells
of
you where you are in the
semi-automatic, so
genome,
we
right?
have
So
to
it tells
hire
you
somebody
gene 1 is
like
here,
Yes.
and it's this
That's
gene
the
is
one.
in gene 2, and gene 3 is in
200
chromosome
an
3,
hour
and
is
gene
what they
10
do.
is in
Is that the
chromosome
120 or
5, and we're
is that
trying to
not
figure
the
out where the
120?
genes are, and by having an
$95,000
inbred
used.
population, and
So
several inbred
the
populations, and we want
600s
these
new
inbred
are
populations,
like
all of them are
$500,000,
tall,
and
and
I'm
these
like,
other inbred
okay, so
populations,
we
all
don't
of
have
them
that
are
budget.
very
That's not
dark
a budget
green,
we have.
and these other ones, all
But
of them
that
have
being said,
very broad leaves. that would help us put those traits, so those physical characteristics
Yeah.
in the map, right?
Yeah.
So the broadleaf characteristics
Yeah.
is here,
Stuff,
in
they have
this
it, they
part
have
of the
it to
genome, then the tall
their
genes
whole order.
are here, and so that's what we're trying to do, but we need to cross.
I haven't tiered it
Yeah.
yet,
So,
but
So that's
I
sort of
started
the kind of work
thinking.
that we're doing. We're doing phenotype
So-
hunting, looking for certain characteristics of the plant that we like. Whether it
they
is
were like,
yield
hey,
of
our
the
stress all
plant,
being
whether it
delivered
is
for,
pest
and I was
resistance,
like,
whether it is
Exactly.
internodal
In
relationship.
those days, or
their whole
Bud
quality,
height,
or
exactly.
high
So I look
CBD,
at this and I'm
Absolutely.
like,
That's the
okay,
kind of
this is
stuff that we're
fertile
doing.
weight,
Now, we're certainly not doing it
all
at your
that.
league
But
level.
they can do
We're
900
just
of
doing
those.
the
And
basic
I know it's not ideal.
plant crossing, looking
If
for
we were
the,
doing
just the way humans have always done, right? Looking for the best
maybe in
plant.
the refuse market, this is the
And
way to go.
so you
Not them, but this.
I have a team there.
Uh-huh.
Yeah, and so they do this work.
Because maybe we can meet and talk about it, because I'm really interested in
Yeah.
establishing a
Right.
collaboration, and I'm here,
Yeah.
I got her.
But
Well, tell you what, you want me to send you an
simplicity
email
of
with my
this
contact
and the
info on
fact
it?
that, okay,
That would
we
be
can do
great.
200
Yeah.
And then maybe we
200
can meet at some
capsules.
point, because I'm
And
really
let's say
interested
this
in
is
trying to
100
pursue this.
doses.
Yeah.
I'm trying to
No,
grow
we have
these
to buy, we
seeds,
have to buy
and
the
we have
line
the
and all
seeds, and
that.
I'm not
But
being
look
able
at
to
that, that's
figure
what,
out where
you
to
know,
grow
an
them,
average manufacturing is
and
you
also,
can do 900,
yeah, I'm interested
so
in,
it was,
in learning more about what
up
you
to
have, which could be really
15.
complementary to what we
Yeah.
have.
No, I
And this is
obviously-
900 per hour.
Absolutely.
No, it's
Okay, so we switched over to
7,200
seeds
in an
about
8-hour
There's
show.
no
6
doubt
months
that'll give
ago.
us better
Now,
I'm
options.
just
2
trying to
million
look at
I'm just
seeds.
saying on what cost.
So this plant is,
And
And if it's
it has a
not
gift for creating seeds.
well, it's a matter of like,
So,
man,
yeah, okay, sure, I'll send you an email, and I'll send you some pictures of it.
Well,
My name is Rich.
Oh, it's
Rich
not.
Max.
Of course.
Well,
I
we've
know,
got easily
not
been
good.
talking about